All right. Good evening, Dr. Ricordi.
Good evening, Sean.
It's great to see you. I want to welcome everybody who is joining us tonight for this really special occasion. We're thrilled to have Dr. Ricordi as our Meet the Scientist for tonight. And we have guests joining literally from all over the world, including Egypt, India, England, Italy, and Asia, to name just a few, as we know that diabetes research is a worldwide effort and affects people literally from everywhere across the globe. I hope tonight that you'll find this session to be both informative and be inspired by the critical work that we are doing at the Diabetes Research Institute right now to cure diabetes. Just a brief overview of both the DRI and the foundation. And the DRI leads the world in cure-focused diabetes research, and as one of the largest and most comprehensive research centers dedicated to curing diabetes, the DRI is aggressively working to develop a biological cure for diabetes by restoring natural insulin production and normalizing blood sugar levels without imposing other risks. The mission of the Diabetes Research Institute Foundation is to provide the Institute with the funding that is necessary to cure diabetes now. And we thank you for the critical role that you play in curing this disease. The groundbreaking work at the DRI and beyond would certainly not be possible without supporters and donors like you. So we're thrilled to have you here. And this is truly a special occasion because we get to be up close and personal with our scientific director of the Diabetes Research Institute, Dr. Camillo Ricordi. So the session will consist of questions that I will be asking based on your feedback. Many of you that submitted those questions when you registered. And if there's time at the end, we will open up to some additional questions that were submitted throughout the session through our chat box. So please make sure that you're taking a look at the chat box and ask questions that you can. We cannot promise that we'll get to every single question that's asked, but we will try our best. And we're hopeful as well to be able to answer some of those questions as a follow-up piece to the recording. That being said, the session is being recorded and we'll release it at a later date that you'll be able to both share, review, or go back and take a look at once the evening is over. So I am pleased to have the honor of introducing the distinguished Dr. Camillo Ricordi. And Camillo is the Stacey Joy Goodman Professor of Surgery and Director of the Diabetes Research Institute and the Cell Transplant Center at the University of Miami. Dr. Ricordi is a world leader in diabetes cure-focused research and cell transplantation, and he invented the technology to extract large number of insulin-producing cells from the human pancreas and perform the first successful transplant to reverse diabetes. His research interests include the reversal of autoimmunity, immune tolerance, stem cells, and regenerative medicine. Dr. Ricordi and collaborators developed strategies to treat type 1 diabetes and other diseases using umbilical cord-derived mesenchymal stem cells. And recently, Dr. Ricordi and his collaborators have used these cells to save patients with severe COVID-19 and diabetes. So Camillo, thank you so much for being here tonight and sharing your exciting work with us. I'm thrilled to be able to welcome you. So let's start off with, why don't you tell us a little bit about yourself for those of you that are not familiar with you?
Yes, well, since we have a restricted time, I just put my Instagram link and Facebook and LinkedIn link and Twitter. So if people want to know more or keep more in touch, I just want to tell you, I don't answer questions on LinkedIn because it's over 10,000 people, but I try to answer them on Instagram or occasionally on Facebook. But I think it's fantastic that you're all. I'm sorry for the time in Italy or East that is more. It's like a little late in the night, but I hope I will be entertaining enough to keep you awake for a little while. So, Sean, what was your first question?
So tell us a little bit about yourself.
Well, I'm from Italy. Actually, I was born in New York, as you can tell from my accent, but I actually left New York for Italy when I was 3 months old, grew up in Italy, did medical school in Italy, Milan, and then moved to Washington University in St. Louis, and then University of Pittsburgh Transplant Institute, and I've been at the University of Miami since '93. I used to have hobbies. I used to have been involved in extreme sport. I was a downhill ski racer, windsurfing races, park motorcycles, and then my mother told me when I started medical school, said, "Are you sure you can finish the 6-year medical school in Italy? It's very competitive, and you should have a plan B." So I became ski instructor and windsurfing instructor just in case I couldn't finish medical school. And now I don't do any more extreme sport. My major hobbies are to explore the unsolved mysteries of the universe and connecting the dots between the major pandemics of the 21st century, which are autoimmunity like type 1 diabetes, COVID-19, and unhealthy lifespan, age-related diseases that they have all in common links and risk factors that can be overcome. And also the only sport that I do very actively is playing ping pong with my new wife, Mi Wang.
But I understand that you are an accomplished chef. So if you were going to cook your favorite dish, what's your best dish that you would cook for us tonight?
Well, it would depend if there are more than 100 people or less than 20, but one that could go for all would be something like in line with my anti-inflammatory nutrition programs, like a little glycemic, a little low glycemic index pasta, like the Barilla collection pasta that is my favorite, with a saltfish cubes, cherry tomatoes, and mint. That is very healthy and has your dose of omega-3, or a little of the omega-3 you should get.
I love that. I'll be right over for that dish as soon as we're done tonight. So, on to more serious discussion regarding diabetes. So from your perspective, what makes the Diabetes Research Institute unique compared to other research institutions?
Well, what makes the DRI unique besides the fact that I'm directing it, and that is critical to keep the mission focus and the Institute philosophy and strategy focus on curing diabetes in the fastest, most efficient way possible. I've been fortunate to be able to assemble an amazing, dedicated team that has delivered incredible progress even during this unprecedented pandemic. So I think it is really a unique place, not just for the team and the fact that I'm leading it, but is the strategy and the philosophy is like you have to share everything you know with the rest of the world. You have to collaborate, you have to be open and have the same intensity of helping your own scientists within the Institute and people anywhere on the globe that want to share our mission.
Which I think is great. And for those of you that are joining tonight, if you're not familiar with my own story, I was diagnosed as a diabetic now about 5 years ago. And I'm a 4th generation person living with diabetes in my own family. So it's personal to me. And I'm also a patient at the DRI. I think one of the interesting things that I noticed, and if you could comment on, is the actual architecture of the DRI building itself, the way that it was designed to make sure that, the first floor is for clinical. But could you, talk a little bit about the uniqueness of the architecture of the building?
Yeah, well, the unique architecture is because we have a research tower that is the cell transplant center that houses the heart of the DRI, that is the CGMP, that means current good manufacturing practice facility for advanced cell and biologic manufacturing for the clinical trials. That's where everything we produce from the islet, from the stem to the stem cells are done on the 4th floor. But the unique feature is that all the scientists, we have over 120 people working in the Institute, when they go to the research tower, to the cell transplant center, they have to cross the lobby with the patients in diagonal, so that is a subliminal message to remember that they're there to cure diabetes in the fastest, most efficient way possible. It's not an exercise, it's not for fancy paper, it's not for Nobel Prizes. Is with a mission and a promise that we made to cure diabetes in the fastest, most efficient way possible. So even the building has been designed to remind everyone every day, every morning, of why they're there and what is the mission.
And we even have some of our researchers that are personally connected to type 1. Can you talk about those?
Sure, like we have many people, like starting from my former executive assistant that now, luckily for her, retired at age 70, Mabel Lewis, has been an incredible helped through the 3 decades I've been at the DRI, and she had her daughter, her husband, and her mother with type 1 diabetes. So she's been in charge and keeping me on track on what is important, what is relevant, what is not, and pushing me every day and give extra motivation. We have also Norma Kenyon had a daughter that was diagnosed. Chris Frecker has type 1 by itself. We have so many people that have either been impacted personally or in a child or relatives, so that you feel the urgency throughout the institute. When you walk the DRI, there is no one that is like just casually there to do some kind of scientific enterprise.
Yeah, and I appreciate that, especially hearing, folks that are living with this disease and impacted by it. We all have a personal stake in this for sure. So can you share with us some of the most exciting things that are happening at the Institute right now in the laboratories?
Well, it has been really an exciting year, despite this unprecedented pandemic that has crippled a lot of the ability to be more and more effective. But we had the most spectacular year as far as advances towards clinical application, new breakthrough. We had the analysis of the 20-year survival data after islet transplantation that showed that islet transplantation, despite the use of chronic immunosuppression, has a significant advantage over insulin without immunosuppression when you do the transplant in an age range around 40, 45 years of age. You wouldn't do a transplant with immunosuppression in a child, but if you are above or over 40 years of age, even with immunosuppression, which seems to be, if not superior, at least equal to insulin treatment, and has been really a remarkable advancement, because in the past, endocrinology and diabetologists will say, no, transplantation cannot be done because you need immunosuppression, it's too dangerous. Well, we now show that you can have 85% 20-year patient survival with immunosuppression and islet transplant, while you have less than 60%, what is reported in the literature with diabetes and insulin without immunosuppression. So puts that islet transplantation could be even considered a life-saving procedure in certain age populations. And in addition to this, we had this incredible results of umbilical cord mesenchymal stem cell infusion. For those of you who don't know, mesenchymal stem cell a form of stem cell that can be obtained by the umbilical cord, that is after every baby is born, healthy newborn baby, generally discard the placenta, the perinatal tissue like the umbilical cord. Some people store and bank the cord blood, but the cord, the vessel is thrown away and it contains an incredible power of regenerative and stem cells that can be extracted from a so-called subepithelial lining, but from the lining of the umbilical cord. And they have such an incredible power to expand and promote immunomodulation, anti-inflammatory, promote tissue regeneration and repair. And from a single baby newborn umbilical cord, you can generate over 90,000 therapeutic doses. So if you think that in organ transplantation, we have the problem because of the lack of donors in the United States alone, we lose over 11,000 patients die every year because of lack of donor organs. Here you have a source of stem cell that could save lives and be used for many therapeutic applications with unlimited potential, because if every baby can save 90,000 people, there is sure not a shortage of newborns. So it's a very exciting source. And we have now a paper that is now in press. You will read it very soon in cytotherapy, that show that umbilical cord mesenchymal stem cell infusion in subject with long-term type 1 diabetes were able to block the progression of complication of diabetes, like nephropathy, neuropathy, retinopathy, like kidney disease, eye disease, and neural disease. And with an 8-year follow-up in a randomized trial, so a very rigorous trial. Also, Dr. Kenyon team showed that mesenchymal stem cell can prevent rejection of islet transplant and substantially can potentiate the survival of islet transplant. And I can give you the news that we just got approval yesterday from the Food and Drug Administration to start the first patient of what hopefully will become a larger trial of umbilical cord mesenchymal stem cell infusion to reverse diabetes nephropathy, like diabetic kidney disease, and prevent dialysis and renal transplant requirement in patients that are starting to show sign of deterioration of kidney function. In addition, we are collaborating with a lot of companies, startup, and new potential breakthrough for type 1 diabetes cure. And then I can list them all, but will take a couple of hours. But the most exciting we can identify, like in eye tolerance, is a new collaboration to define strategy to do transplant without immunosuppression. And we already have data that are submitted for publication. The most exciting publication I saw in the past 20 years, where you can have islet transplant functioning for months without immunosuppressive drugs after a very short time of immunomodulation at the beginning of the transplant. And if you think what you probably have seen from the report from Vertex and ViaCyte on stem cell derived islets to address the problem of the shortage of insulin producing cells that could be obtained from organs. Now we may have strategies that address the unlimited source of insulin producing cells, so being able to have as many cells as we need to treat all patients with diabetes on one side, and then strategies to induce tolerance without immunosuppression. So if you combine the two, could be the real holy grail of what we've been trying to do for the last 3 decades.
Which is phenomenal. So along those lines, when you're speaking to scientists and researchers, nationally and internationally, what are some of the things that you're sharing when you address the work and in terms of what we're doing towards a cure at the DRI?
Well, they know, they generally contact us because they know that we're well aware that what distinguishes us, besides the leadership and the multidisciplinary expertise that allow us to help efforts worldwide, no matter at what stage of development they are, it is our mission-driven sharing collaborative strategies that distinguish us, and that is why many, from big pharma to new startups, from major institutions like Harvard, Johns Hopkins, Stanford, from China to Europe, from Canada to South America, we are willing to collaborate and help anyone with it who has a serious commitment or a breakthrough idea on the path to a cure for type 1 diabetes.
And that's one of the things that I love about the Institute is that for us, and even on the foundation side, it doesn't matter to us where the cure comes from, as long as a cure is found, and the idea and the notion of collaboration, I think, is going to be critical in that advancement. So in the area of immunology, cell transplantation, cell research, I know you talked a little bit about that, but what are some of the advances that are being made, and what are you most excited about in those areas?
Well, the thing that I'm most excited is the potential synergies between the identification of stem cell derived islets that will overcome the limitation of organ donation, like the Vertex trial. I'm sure many of you have heard or seen or read the press release. There was the first patient transplanted. We've been the first center activated in the trial together with Harvard, Penn, and now there are many more centers joining this trial. Is the first proof of moving from islets extracted from donor organ pancreas to an unlimited, potentially unlimited source of stem cell derived islets that will eliminate the need for organ donation. And ViaCyte presented a few weeks ago their first patient. Was very encouraging because it has some C-peptide production, not much insulin reduction in the requirement, but lowering of hemoglobin A1C, and was very promising. I make very promising and optimistic comments. The Vertex announcement has been mind-blowing to me personally, because it had the same production that we can expect from a successful islet transplant, despite they use half of the dose, because FDA for the first 2 patients required 50% dose. And I had the honor and the privilege to work with Doug Melton and the Harvard Stem Cell Institute team previously Sema Therapeutics and now part of Vertex on this technology for several years. And I think is spectacular that you can say that what was an idea and results in preclinical model system is now giving the success that they recorded in the first patient in a clinical trial. Besides, so this says if we have an unlimited source, then the key is how do you do this transplant without immunosuppression? And the fact that we have strategies like high tolerance, strategy to eliminate immunosuppression within weeks and then have islets surviving for long term without any immunosuppressive agent, any anti-rejection agent. Or we have also other collaboration like with Eledon that was an elixir with anti-CD40 ligand that is another powerful immunomodulation agent for immunomodulation that will replace Prograf immunosuppression, and trials are just starting now is amazing. We have also so many collaboration with European entities that produce other forms of stem cell-derived islets. We even have a collaboration with California and Mexico on a technology to do a gene therapy to a segment of the liver and convert it into an endocrine pancreas without immunosuppression. We have trials that hopefully will start next year with Alberto Pugliese and collaborators on low-dose IL-2 to try to stimulate regulatory T cells. We have a collaboration with Poland that they develop a method to expand regulatory T cells that are the ones that keep the balance between autoreactive, the cells that try to attack and kill the islets, and the one that regulate them and keep them autoimmunity on check. And they already have results in clinical trial with 5 years follow-up, being able to maintain insulin production in children and young adults that undergo this protocol without losing C-peptide production. We also have the DRI new trial using DMF, that is dimethyl fumarate, which is a drug that has been approved already for treatment of multiple sclerosis by FDA, and is showing potential also for treating autoimmune diseases such as type 1 diabetes, because it has an antioxidant and anti-inflammatory action, and could be synergistic with many of the other strategies that we're testing, like the vitamin D omega-3 Poseidon trial that we've been testing to try to halt progression of autoimmunity by immunomodulation without immunosuppression at the beginning of the disease. And unfortunately, we were halted because during the COVID pandemic, many of these trials have to be stopped and stuck. And so now we're being able to resurrect all of these technologies with new ones that are emerging, like combination of polyphenols and sirtuin activators that like SIRT-500 plus, that is a new agent that has been studied together with centers in Europe, and that was born as anti-aging, but now turned out to protect against COVID and has also effect on type 1 diabetes, with some of the patients on this call that already experienced the beneficial effect of this combination with vitamin D and omega-3.
So there is certainly no shortage of work that's going on, and I get excited when I hear, between Norma Kenyon working on her anti-CD40 ligand trial, or Alberto Pugliese with his IL-2 trial, just an amazing amount of work and excitement that I certainly can see from my perspective on the outside. But talk to me about when a family with a loved one visits the DRI today. What do you think is different today than if they had visited 20 years ago?
Well, everything pretty much, because 20 years ago we were talking about promise, we will cure diabetes, it's not a prediction, it's a promise kind of approach. And we said everything. We have also promising strategies in small animal model, mice and others. But unfortunately, what we painfully became aware is that there have been over 440 strategies that were successful in mice, and none of them translated to an effective cure in type 1 clinically. So that's why our effort has progressively shifted more and more and more towards okay, you have an early data in a preclinical model, let's do the pilot clinical trial and show if it's significant or relevant to cure patients. That is our objective. We don't want to get another 5-year grants and another renewal of an NIH grant to study more and more and about less and less in an irrelevant model system. So that has been what I can see is that what we were talking about, hope and promise and what could be promising back then, now everything is getting to the clinical trials. I've never seen in the last year, we're submerged, we're like paralyzed by this volume of things that we could do and that we don't have enough funding to do or not enough infrastructure, and how my policy and strategy of working completely in the utopistic non-profit arena, saying we share everything, I give up all my patents or my invention to scientists worldwide, and then at the end, nobody's going to give you the hundreds of millions necessary to bring a new therapy from an idea to actual delivery to patients. And I'm not talking the phase 1 trial, pilot trial. I'm saying how do you do the validation trial, the phase 3 trial, the infrastructure that is necessary to bring a new therapy to a real cure.
So, which is great, and totally understand that perspective. So, in terms of hope, it's a big word, and I know that coming into the diabetes community, I have heard from a number of folks that said, well, I was promised, in 10 years I was going to have a cure for my daughter, right? And science moves slow. We know that. Obviously, there's a lot of things that are progressing fairly rapidly right now. So without giving a timeline, because I think it's a fool's errand to try and do that from a scientific perspective, what are you most hopeful about right now in terms of where we are headed towards a cure?
The risk of being hit tomorrow by critics, like I used to say, the cure could be 5 to 7 years or more than 10 years away. But now with what I'm seeing emerging and moving to clinical trial, I wouldn't be at all surprised if it could be something in the next 3 years. And so I'm not promising a cure in 3 years, but I'm so excited. I see so many breakthroughs recently and so much moving to clinical trials that if we have an unlimited source of stem cells, like I don't want to keep citing the same companies, but if you have resolved the problem of supply, because if we have a successful biologic replacement strategy, you would be stuck with islets from organ donors. So you can get 1,000 pancreas a year in the United States. You have 1.6 million patients with type 1 diabetes. Worldwide, you have 26 million. So it will become a lottery to say who can be cured. Now, if we have the unlimited source of insulin producing cells on one side, and if you have a successful strategy to eliminate immunosuppression, this is the holy grail. I can go cure another disease in 3 years. So all of this is moving to clinical trials. So we've been there before. We cannot be over-optimistic. We cannot throw false hope in patients and family. But I personally have never been so excited in my entire professional life. And I really think that this gives the energy to keep concentrated, keep focused on a cure, and don't get distracted by people or diabetology, or people say, you have diabetes, you have to live for the rest of your life with that. Give up any hope or any false hope on a cure. We are going to get this done, and it's happening beyond our eyes. And I think it's so amazing to be part of this team and this town and this DRI and the foundation and all our collaboration worldwide, because these are the teams that are making this happen.
Which is just amazing. And I tell people that I meet on a pretty consistent basis, and my team will vouch for this, that I have one job, and that's to lose my job. And I'm excited about what I am both seeing and hearing. So along those lines, why is the foundation such an important partner to the scientists and to the Institute?
Well, the foundation has been the critical element in anything that we've been doing, because it gives you the resources to develop your ideas and to give off the starting blocks. So you can say, okay, you have an idea, you want to do a pilot trial, you want to do a new preclinical model, you want to test a breakthrough initiative, you want to develop the core infrastructure to help patients and help groups worldwide, whether they come from commercial or from academic institution, whether they are from Stanford or Harvard or Johns Hopkins or from India or China or Italy or South America or Canada. We help everyone, and that would not be possible without the DRI Foundation, because in a regular institution, you go there, say, can I collaborate with you? And the first thing they ask you, yes, show me the money. How much do you have to support this idea? So we have the ability to do screening, but hands-on due diligence. Do you have an idea? We can test, is this really a potential breakthrough? If it is, we help you with the same enthusiasm and intensity as it is our intramural program or one of our scientists. So that's what distinguishes us and what could never have been done without the Diabetes Research Institute Foundation. And then we have this dual competition between the two organizations in a way, because we keep each other in check, because the DRI Foundation helped the DRI because we are the best hope for a cure, and the DRI keeps in check the foundation because then you have the best hope to fund the cure. If one or the other falls short in their mission, then we can. Our only loyalty is to the cure, is not to any team.
I love that, and we are completely aligned in that regard, right? Our only loyalty is to get to the end goal, which is that cure. So if you could pick one thing to change or accelerate the journey towards that cure, what would it be and why?
Besides funding, I mean, like scientifically?
Besides funding, yes.
Well, scientifically is now the ability to get away from chronic immunosuppression to have a successful biologic replacement on one side. And we're getting there. If this initial work and data that we have from intolerance, Eledon, mesenchymal stem cells, or potential combination of this technology, I think we will get out of the need of chronic immunosuppression very soon. Then we have the other thing is how to prevent recurrence of autoimmune disease and how to prevent recurrence of diabetes once you cure it. And then is where all the trials like with DMF, vitamin D, omega-3, SIRT 500 plus, how you can reestablish a tolerance environment that prevent triggering events that are so critical to develop autoimmunity or COVID-19. And we learned so much from the COVID-19 pandemic because we were able to confirm our theories with these trials. And I think, and then the last one is the unlimited source of insulin-producing cell that is really the key. So they all go together. You cannot have an unlimited source of insulin-producing cells if you don't have tolerance, because then you're stuck with a limited number of patients you can help if they have enough risk to justify immunosuppression. Then you don't need the stem cell because you can do it with 1,000 pancreas a year that we have in the United States. So, but besides stem cells, we are working also and collaborating with groups that are doing gene editing of pig islets so that you can edit pig islets to become less immunogenic, to enhance less rejection than human islets. And so that this could be another source of insulin producing cells. If you think that unfortunately for the pigs, there are slaughterhouses producing like 60 to 70 million pigs in the United States just for pork chops. If you can cure diabetes in the meantime, instead of throwing away that pancreas, is probably nobody would complain that much. Still, some will complain.
Yeah, certainly. So I want to get to some of the questions from the participants that we have. And we have, again, participants have been, you all have been amazing at posting and asking some questions. So let's get to some of the ones that have come up. One that I thought was interesting is, what would you do with a $50 million gift? Or if money were no object, what would be done with that?
So it's interesting because we just did the strategic planning of what we do, and we said that with 70 to 80 million would be better, but with 50 million, let me scale down my ambition. And this, I think we have three areas that are critically needed. One is the potentiation of the CGMP, of the advanced cell and biologic manufacturing facility to produce clinical grade products that can go in clinical trials. we were lucky enough, thanks to the North American building trades unions, to be able to have an initial grants through the COVID pandemic emergency that allow us to build and buy some of the bioreactor, three-dimensional bioreactors and tangential flow filtration system, and some tip of the iceberg of the research infrastructure that we will need to build to face the need of the new wave of clinical trial. But has been great, and we need to potentiate dramatically the clinical trial infrastructure, like clinical research coordinators, monitoring groups, because the complexity of bringing a new idea to patients, patient may think, okay, you have an idea, why don't you do it? You have to deal with Food and Drug Administration. They ask you for what are your results in small animal models, large animal models, 2 models, 3 models. What is your potency test in development to validate? So practically in cell therapy, we're treated like a big pharma, and the cell product like a drug. So that is like the complexity and cost of bringing anything to patient and crossing the several valleys of death is not just the one from going from an idea to a pilot clinical trial. Is the major value of death is after you're successful in initial pilot trial, how do you bring it to the phase 3 trial, and then to the biological license application, and then to approval, reimbursement, commercialization. So that's why, I thought that, non-profit and utopistic, give everything around for free and don't worry was the way to go. I never charged a patient in my life, and I hope to continue to do so. But the reality is that if you don't team up with the industry in this country, in the United States at least, maybe Europe and Asia may have better possibility, but in the United States, the cost of bringing and the threshold that is keeping raised of what is needed as funding to bring anything to patients is prohibited for an academic institution or for an academic scientist. And then you have the institution that tell you, do you want to become a drug? If you're considered as a drug and a drug manufacturer, the university doesn't want the liability to become a drug manufacturer, because then patient could sue the university or the endowment, if anything. So there is also other kind of consideration that come in place. But resources to build up the infrastructure to be able to move this trial beyond the pilot phase is critically important. And then, of course, we have like 10 to 12 clinical trials in the pipeline that we would need to fund, and that they're all on hold for lack of funding. So make sure there is no lack of ideas on how to use 50 million. Possibly 70 to 80 would be a more idealistic budget a year to move things towards the clinic.
Right.
And it's nothing. This is nothing compared to industry, because you think we spend 65 billion a year in R&D pharmaceutical, maybe to develop a drug that increase the lifespan in a cancer by 3 weeks, or painful 3 weeks, or better GLP-1, a better insulin molecule to put a better band-aid on a wound that never healed. But how much of this 65 billion is spent to cure a disease? Question mark. You tell me. I cannot find even a fraction. And so to think about 80 millions a year to cure a disease that afflicts 460 million of the population in the world and is responsible of 1/10 of the cost of our 3.8 trillion budget in healthcare, that is 20% of the GDP of the country, to me is nothing, 80 million a year. And I wish I will find someone out there that will share my goal.
Well, I think it's doable. That's the number that I've been talking about since I arrived to the foundation. I love to see us even develop a clinical trial fund that would really, be available for all of these trials that are coming in. Because the thing that I want to make sure doesn't happen is that we have an amazing breakthrough that we want to move through to an actual human trial, and we're stuck because of the funding. So, our team is out there every day talking to folks, and we're blessed with an amazing philanthropic community in the diabetes space. We just need to have the conversation and talk about the reason why that, that 60 to 70 to 80 million dollars annually is important to get to our end goal. Completely agree with that.
And I see that, there are people out there that would be like offering a coffee for us to chip in a 60 to 80 million to cure a disease that kills one patient every 7 seconds. And I noticed that there is Zuckerberg and Bill Gates on the webinar. No, sorry, it's too many names. But if there are like someone with this kind of contact or potential, besides changing the name of Facebook to Meta, another very fantastic initiative, help us cure a disease that afflicts 460 million, and we'll be forever grateful.
It certainly makes more sense than flying off to space. So another question that came in is, what has COVID-19 taught us about diabetes research?
Well, a lot. Like it's been, we've been practically, you take someone a little crazy like me and with a background of transplant surgery to say you have a challenge, an epidemic and a paralysis of the system, we just deal with it, come up with a solution. And our solution was that we were already approved to do trials in type 1 diabetes and in Alzheimer using this umbilical cord mesenchymal stem cells. One of the problems of these cells that are immunomodulatory, they fight inflammation, and they fight the cytokine storm that you have in COVID or in autoimmune disease at the onset. The problem is that they go to the lung if you inject them IV, like a blood transfusion. So in diabetes, you would have to infuse them like in catheterizing an artery, femoral artery, interventional radiology, more complicated to get to the pancreas. But when COVID exploded, we said we already have the cells that are approved by FDA, and we raise independently from the foundation, because we don't want to use one penny for diabetes cure to another disease, but thanks to the Cure Alliance and friends and then NAPTO, we were able to do this trial that was approved by FDA in 1 week, and we were able to save 100% of patients of age less than 85 years and 91% of all ages, while unfortunately only 42% survive 1 month in the control group. And more importantly, 100% of subjects, they were almost all with diabetes, that is a comorbidity of COVID, and we save 100% of patients with diabetes. So this was incredible. Our team was so happy to be able, in a crisis where all our trials were blocked because you couldn't do during COVID, you couldn't do an islet transplant, you couldn't do the Poseidon trial, you could not receive a patient in the diabetes, but we were able to save life even during a pandemic and an emergency. Besides the fact that I got COVID twice while doing that, but I survived.
And we're very thankful for that. Yeah,
comorbidity, but the most important is that this trial show and confirm the hypothesis that these cells are immunomodulatory and anti-inflammatory. So when we initially proposed it for type 1 diabetes, people would say, what data you have to demonstrate that this will have an anti-inflammatory immunomodulatory effect? And now we have this data and the safety data of this patient and the clinical data, the mechanistic data. So now we're going back with new trials, and the fact that FDA has approved yesterday the first patients with type 1 diabetes, who I think is on the call tonight, that is coming to Miami next Sunday and will be treated Tuesday and Thursday, is amazing, because we would not have been able to start this new diabetes trial without the data from the COVID trial. So everything is falling in place, but despite the COVID trial and despite the COVID pandemic, I couldn't be more proud of our team for the amazing progress we have done despite an unprecedented pandemic.
I couldn't agree more. You know, it was terrifying going through the pandemic, and you know, every time I would listen to the news, you always heard the comorbidities of the three, right? Asthma, obesity, and diabetes. And so
hypertension, yes.
Yeah, it was terrifying. So the fact that we were able to work through that, you know, we had our tagline that we utilized throughout the pandemic was "Hope isn't quarantine," because our scientists, as you were in the labs every day, still going to work.
But you know, another very interesting thing has been that besides saving the life of people in ICU intubated with the same strategy that could work in type 1 diabetes to block autoimmunity, we said, wait a minute, maybe the same strategy we're planning to use to block autoimmunity, like omega-3, vitamin D, CYRT 500 plus, could work also for COVID. And in fact, we treated over 100 patients. None of them progressed to a disease or symptomatic, and they all became negative again within a week. So this shows you connecting the dots between these pandemics, that they all have an inflammatory immune activation base, that the same strategy you use for autoimmunity may help COVID and vice versa. So it's been very exciting, despite the painful reality of the pandemic, to see how actually new ideas and breakthrough can even emerge in a crisis like this. So,
yeah, certainly. So I want to bring up a question that I see in the chat box, and that it relates to the immune system. They said they hear that a cure is risky because of messing with the immune system and causing cancer. Are some of these potential treatments safe from that risk?
That is a very important question. Like, there have been issue, like if you use stem cells that are brought back to become embryonic, or using embryonic stem cells, like if you use inducible pluripotent stem cells, that is taking an adult cells, making it become embryonic, and then differentiating it into an insulin-producing cells. If in your cell product there are still undifferentiated cells that can become everything, that can cause cancer. So that has been something, and the reason why many of these trials have waited so long, because we have to document exactly there are no cancer potential inducing cells, there is no change in karyotype, there are all these massive tests, years of tests, and hundreds of animal tested before they go into a clinical trial. Still, unfortunately, there are centers in obscure countries, which I will not mention, that offer stem cell therapies for diabetes, and people go into this country to get unproven, unqualified, uncharacterized cell products. And I just spoke with a colleague of mine in Europe that was reviewing a paper from one of these countries in Asia, saying a patient died of teratocarcinoma, that is a terrible malignant cancer, because they had the idea to use his own cells, make them inducible pluripotent stem cells, so go back to embryonic, and then infuse them back in the patient to treat him. But now guess what? Now these cells are not allogenic, like in the case of other trial by or vertex. They're your own cells, so nobody's going to reject them. If you develop a malignant cancer of those cells, you're not going to reject it. And so it's such a critical step that you have to consider, because it's enough to have a handful of wrong cases done by the wrong people, and the whole field goes back 20 years. And then I cannot keep my promise.
Right. So you mentioned working with other institutions. What are some of the most exciting collaborations with other researchers and institutions that we have right now?
Like we have such, how many hours do we have left? So we are working like our traditional best collaborators in the DRI Federation. As you know, we have a Diabetes Research Institute Federation that includes all the centers worldwide that are willing to work with us. And so from China, who was instrumental to start all the umbilical cord mesenchymal stem cell trial in kidney transplantation before, then in diabetes, now the 8 years follow-up results, and now the COVID trial, even the initial results came from them, has been a tremendous collaboration. We had from Italy, historical big collaboration with Italian centers. We are doing now with Niguarda and Federico Bertuzzi and Riccardo Calafiore, the first trial of encapsulated islets, human islets, without immunosuppression. There should be a patient transplanted any week now. We just had an exchange of communication on how to do the next trial using a modification of the biohub strategy that we developed to transplant cells in the omentum. We have such exciting interaction, collaboration with the Vertex investigator for the stem cell-derived islets, Harvard, Penn, Chicago, many centers now worldwide, also in Europe. We have the high tolerance collaboration also with Harvard and Georgia Tech and Missouri and Europe to bring this technology without the need for anti-rejection trial drugs. We have Unisite collaboration with Turin University, and very exciting new strategy with stem cells that could not only generate islets but also hepatocytes to cure liver disease. We have a collaboration with the Polish group and Piotr Tornowski and Paul Tirek that already have the trials done in Europe for regulatory T cell expanded by the patient own blood and reinfused to block progression of type 1 diabetes with 5 years follow-up results, maintaining C-peptide production without immunosuppression. We have the gene therapy localized to the liver in collaboration with Mexico and California, that we hope to find the resources to bring it forward and convert a little piece of the liver into an endocrine pancreas. We have the work with Eledon and the collaborators of Eledon with the anti-CD40 ligand, bringing forward the credible work of Norma Kenyon and the entire team to introduce co-stimulatory blockade as a strategy to replace standard immunosuppression. Also very exciting. And then we have all the mesenchymal stem cell work with people worldwide and centers that there are at least 200 trials ongoing with mesenchymal stem cell now, and not just in diabetes and autoimmune diseases, but also for COVID and other chronic degenerative disease condition. And I think a combination of this or one of these will be the winner. But we are in the final, we're in the World Cup zone. It's not anymore like fishing or saying, oh, let's guess what could emerge. They emerge and there are the finalists, and the finalists are less than 12, and may the best win.
Absolutely, absolutely.
With our help or without, but we'll help anyone.
Certainly. So I want to bring up one of the things that you mentioned is other autoimmune disease. There was a question that came up that said, is any of the work that we are doing at the DRI eventually transferable to other autoimmune diseases? Obviously, we know things like celiac disease, right, impact those living with type 1 at a pretty significant rate. So
yeah, it is a critical question, because actually we think people used to say, why you work with type 1 diabetes is only 5% of diabetes total in the world. Why don't you work on type 2? Because actually type 1 diabetes may be the tip of the iceberg of the autoimmune pandemic that is affecting 20% of the population with 100 different disease conditions, of which type 1 diabetes is one. But what you develop to cure type 1 diabetes could help all other autoimmune diseases, or the majority of them, because it's a rebalancing the immune system, restoring tolerance. Autoimmunity means that your body loses the ability to protect his own tissue and goes crazy and start attacking cells of your own body, not recognizing them as self. In type 1 diabetes, it is the beta cell insulin producing cells, but you can have it in lupus, scleroderma, the skin and other organs. You can have a vitiligo, psoriasis, literally 100, kidney disease, liver disease. But the interesting thing is that the strategy we're working for type 1 diabetes may help other diseases. Like, for example, now we are working with this combination that is vitamin D, omega-3, and SIRT-500 plus in diabetes. Then I get calls from desperate friends from Italy or Europe saying, I was just diagnosed with a liver autoimmune disease. I have to go start immunosuppression next month. And I said, well, before you go next month, why don't you try this protocol? It's safe. There are natural substances that are generic. You can just get them on the internet. Try. If it doesn't work, sorry. And then she calls me back crying, saying, I went back. They says, I have no sign of liver autoimmunity. We have so many of these anecdotal cases, but it would be so great to be able to do a formal validation, clinical trials, and find a few $1 million also to treat these strategies. But the fact that they're generic drugs and natural substances, and there is no $1 trillion to be made profit using them, nobody wants to fund this kind of prevention trials or using simple molecules, natural molecules. So that's another area where the foundation could help a lot.
So I know we just have a few minutes left, but I like this question that came in on the chat line, because in the work that we do, we come across people that have been diagnosed for 40 years, 45 years. I was with somebody today at lunch, 26 years. And the question is, what are the possibilities of some type of help for those of us who are long-term T1Ds and somewhat aged?
Well, is even more important if you're long-term T1D, everything we're trying to develop, like well, first of all, there is a whole areas, wellness medicine and health span medicine that interface with diabetes, because diabetes is an accelerated aging disease. We have to face it. Unfortunately, in diabetes, like there was a Lancet article that clearly defined that if you are diagnosed with diabetes before age 10, you have the probability to lose 2 decades, over 20 years of life if you're up to 20 years of life, if you're a woman, up to 18 years of life if you're a man. But it's clearly an accelerated aging disease. I don't want to scare anyone, because now we will have strategies to prevent this accelerated aging, because this accelerated aging is determined by the fact that you have an inflammatory condition underlying that consume faster your stem cell regenerative potential, repair mechanism. So you don't feel anything until you're 40, 50, 60, and then bad things start happening. So now the strategies that we're using, or that people are developing in anti-aging or to fight age-related chronic disease condition, from omega-3, vitamin D, CYP 500 plus, fisetin, there are trials at Mayo using fisetin, an anti-senolytic drug that incidentally has been working also in COVID-19, because it's all a link. Like if you are aging faster, you're also more exposed to COVID. That's why diabetes may be a comorbidity or other age-related disease condition, or age itself. So, you know, I think there is a lot of hope for people that has long-term diabetes. I would start taking, you can check, I have a website that is called Fit for Pandemic, FIT number 4 pandemic, that defines some of these natural strategies that could be used both to prevent autoimmune disease, or to fight age-related chronic disease condition, or to fight COVID. What I define the 3 major pandemics of the 21st century are, if you wish, COVID-19 is a pandemic, but then you have autoimmunity, is another pandemic affecting 20% of the population with 100 disease condition. And then you have the third huge pandemic is age-related chronic disease condition, or unhealthy aging, that is responsible for like 90% of the healthcare expenditures in these countries related to chronic disease, age-related, that in large part are preventable. And if you just prolong by 2 years healthy lifespan, you can save more than $7 trillion to the healthcare expenditure that can be used for curing other diseases of global warming or your favorite cause.
Wonderful. We are coming to the end of our fireside chat tonight. I want to give Dr. Ricordi the last word. I do want to, again, acknowledge everybody who has participated from really across the globe tonight. And we appreciate everybody participating, and your support and your questions are great. We'll try and get to as many of those questions as we are able to. You know, as a follow-up to this, I know there was one question regarding how people can get involved and how they can donate it to the Diabetes Research Institute Foundation. I would encourage you to visit our website at diabetesresearch.org. There is a link there to make a gift. We appreciate every gift that comes in. It all goes towards one goal, which is, as we've been saying, finding a cure for type 1. And we encourage you, if you're not part of our DRI Insiders, to equally sign up for that. Follow us on social media. And with that, I will turn it over to Dr. Ricordi to close us out for the evening.
Yeah, thank you, Sean, and thank you all, for especially all of those who stay up so late from Europe or other countries. And I'm sorry we have limited time tonight, but I also try to put anything that is new or relates to our work. I also put it on our social media, on Instagram, Camillo Ricordi, MD Official, or Facebook, LinkedIn, or Twitter, so that I cannot answer all the time the tens of thousands questions that come, but they're often very similar. And that's why I try to be on the social media and post anything new that is emerging. And I'll try to continue to respond to all. I know there are some that have questions on vitamin D and omega-3. I hope you will be able to see some of this trial also online on the social media, and keep up with the progress and keep up with the hope that now is more than a hope and is more and more than ever closer to the promise that we made years ago. So keep up a positive attitude and give us the energy to continue to remain focused on a cure, because it is from patient families and friends that give us the energy to continue instead of taking a couple of weeks holiday and go fishing.
Wonderful. Well, thank you, Dr. Ricordi, and thank you, everybody. Have a great night.
Thank you, Sean. Thank you all.